Okay, so this is the explanation of how I went through mapping the genes of the biotin pathway, the two methods that I used. So, the first method was a little tedious, but it started with navigating to the PubMed website or database and searching biotin synthesis, Escherichia coli, and then filtering to seeing only reviews. And from there, I just clicked on the first link. When I clicked on the first link, there was quite a bit of information. And then when I scrolled through, I saw that this one author, Cronin, J.E., had several papers regarding this. So that would be the best bet for trying to find the correct biosynthesis pathway to find all of the enzymes that I would be looking for in order to see the enzymes in the mechanism from pimioyl ACP to biotin. From there, I went back and made an advanced search, adding Cronin as the author to see if I could find a pathway that was more detailed than what I was finding in some of the reviews. So after I removed the review filter and navigated down the results, I saw this link to this paper, which was in EcoCell. And when I clicked on it, I found this image, this figure, illustrating a very detailed steps in the mechanism and outlining each of the enzymes that I was looking for. So the enzymes I then copied down, so I would have a reference to look them each up, where BioH, BioF, BioA, BioD, and BioB in that order. And you can see them circled in this image. For each of these enzymes, the assignment asks for the gene name, the enzyme name, the EC number, and the protein sequence. So, again, this first method was tedious in that I just went gene by gene using the NCBI database, starting with the very first enzyme, which was BioH. And I searched BioH, E. coli, MG1655, and searched it as a protein, but what came up was the gene. So I clicked on the gene, and when I clicked on the gene, I navigated to the right hand side of the page under related information was protein, as you can see circled there. I clicked on protein, and the very first link was the information needed for the protein. This information, or the information that I needed as far as the enzyme name and the EC number, were down at the bottom under the protein heading, as you can see circled there. So far I've got the gene name, the enzyme name, the EC number, and then I would just need the protein sequence. So if you scroll to the top of the page, you can see that if you click on FASTA, you can get the protein sequence, which is outlined here. And when you compile all the information, this was all the info that was needed for the assignment, and this process just needed to be repeated for the rest of the enzymes, BioF, BioA, BioD, and BioB. And going through all of them through the same method, these are the rest of the information needed for each of those enzymes. The second method was a much quicker method, and you still had to find the enzymes needed. So the initial steps of finding the pathway and determining which enzymes to look for was the same, but then actually finding the info for each enzyme was much quicker using this method, where I just picked one of the enzymes. In this case it was BioB, and searched again BioB E. coli MG1655 in the NCBI database, and then clicked on the first link for the gene, just like the previous method. However, instead of going down and clicking on protein and going that way, I scrolled down and clicked on the KEGG link on the right hand side under links to other resources, as you can see circled there. When you click on that, it brings you to the respective KEGG site for that enzyme, and this gives you all the information for that enzyme that was needed. So it gives you the protein, or the enzyme name, the gene name, the EC number, and then if you scroll to the bottom, it's not shown here, but at the bottom of the page you can actually get the amino acid sequence as well as other information regarding the gene, like the nucleotide sequence, but that wasn't asked for. And then to quickly jump to the other proteins, you navigate to the pathway tab under biotin metabolism and click on that hyperlink next to it. And when you click on that, it brings you to this extensive pathway with each of the enzymes labeled. So from here, you can actually just click on each of the green boxes associated with each of the enzymes, and then it'll bring you to each of their respective KEGG pages. And again, you can get all of the necessary information much quicker. So yeah, that's all of it.